Hello everyone, today we will learn about proteins. So, see, the proteins are required by our body for building muscles, producing hormones, and also making in enzymes. So, these proteins are very much important for our body. Let's see. These proteins are the complex biomolecules made up of amino acids. Like our body is made up of cells, in the same way, the proteins are made up of amino acids. And these proteins are polymeric in nature. This means that they will form polymers, and mostly they form heteropolymers. There is another type of polymer, homopolymer, where the constituents are same, that is known as homopolymer. But here in a protein the amino acids will be of different types, so this is known as a heteropolymer. And these proteins are the building blocks of the muscles, I told you earlier. And there are a lot of amino acids, but we will deal only with 20 which help in making protein in our body. And in a protein, the left end or the left terminal is known as the N terminal, and the right terminal or right end is known as the C terminal. This N and C means nitrogen and carbon. We will see it later why it is named so. See the structure of proteins. I told you that proteins are polymeric in structure, and I will tell you later that there are 4 different types of structures of protein: primary structure, secondary structure, tertiary structure, and quaternary structure. So we will discuss that later. For now, we will learn that the proteins are made up of amino acids joined by peptide bonds, hence also called polypeptides. I will show you how a polypeptide bond is formed or a peptide bond is formed. I will show it later. So to understand proteins, let's first understand the building blocks of the proteins, that is the amino acids. So when we understand amino acids, then it becomes easy for us to understand about the proteins and their structure. So let's see the structure of amino acids. We can understand it from the name of an amino acid that there is an amino group and a acidic group. The amino group is a NH2 group and the acidic group is a carboxylic acid group, COOH group. Here the amino group is bond NH2, this is read as bond NH2, and the acidic group is read as bond COOH, or like this. We can represent it in two ways. So in a general structure of amino acid, there is an amino group, an acidic group, and a variable hydrocarbon group or R group. I will show you later that how one carbon's valency is distributed among 4 different types of groups. And even we will classify amino acids on the basis of number of acidic and amino groups, namely acidic amino acids and basic amino acids and also neutral amino acids. And another thing, that amino acids can be essential or non-essential. Here I have written it. Amino acids can be essential or non-essential depending on their necessity for body. Now we will see the structure of an amino acid. See, earlier I told you that one carbon's valency is distributed among 4 different types of groups. These groups are one acidic group, a variable R group, and one amino group and a hydrogen. That's why this is also known as methylated amino acids also. They are sometimes known as methylated amino acids because one carbon's valency is distributed among 4 groups. So this is amino group, this is the acidic group, and this is the variable R group, depending on which there are 20 different types of amino acids. Suppose this R group is hydrogen. If this is hydrogen, then this amino acid will be glycine, like this. The structure will be like this: NH2CHHCOOH. Here the variable R group is a hydrogen, and this is the simplest amino acid where R is equal to bond H, and this amino acid glycine is optically inactive. Next we will see alanine. Alanine, where the variable R group is substituted by methyl group or CH3 group. Here you can see NH2CHCH3COOH. Here this amino acid is known as alanine. And then comes serine, where the R group is substituted by bond CH2OH, and this is an alcohol group. you can see it. This is the alcohol group, where the R group is substituted by RCH2OH or CH2OH, and this amino acid is known as serine. Now let's see the formation of a peptide bond. Here is one amino acid, this is one amino acid, and this is the another amino acid. Here you can see it is R1 and this is denoted by R2. The OH group of carboxylic acid of first amino acid reacts with the hydrogen of the amino group of second amino acid, and when they react, it leads to the formation of a molecule of water which leaves the reaction, and there is a formation of a peptide bond. This bond C double bond O bond N bond H, this bond is known as a peptide bond, and this is the most fundamental or the most basic bond present between any kinds of proteins or any kinds of amino acids. I will show you later that there are different kinds of bonds like disulfide bond, hydrogen bond, hydrophobic interaction. There are different kinds, but no matter what kind of bonds are present, this bond will be present, and this is mandatory because this is the basic bonding between any two amino acids. Now we will see how the names of all the 20 amino acids can be memorized. Here is a small trick for you all. Imagine Virat Kohli has been given a homework of listening to the FM radio. So Virat Kohli loves FM HW. HW means homework. So Virat Kohli is loving his homework, and his homework was to listen to FM. So remember it, Virat Kohli loves FM HW. And the second trick is why second Q&A GDP. Q&A means question and answer, GDP means gross domestic product. So a student asks his teacher, why you asked me question on GDP on the second time? So he asked, why second Q&A GDP? So these are the tricks, and the capital letters will be denoted by the symbol of the amino acids. We will memorize it right now. See, VIRTKLFMHW. These are the first 10 amino acids, and these first 10 are essential. If you remember this trick, then you don't need to remember about essential and non-essential amino acids. You can keep it in mind. Listen, V for valine, I for isoleucine, R for arginine. Here you can pronounce it like arginine. Arginine, it seems like it's starting with R, so the symbol will be R. Then T for threonine, K for lysine. Just remember this one is the most odd one. Here in alphabetical order, after K, L comes, so K is the symbol for lysine, and L for leucine, F for phenylalanine. It seems like it starts with F. You can see that this F is the symbol for phenylalanine, and when you pronounce phenylalanine, it seems like it starts with F, so its symbol is F. And M for methionine, H for histidine, and W is for tryptophan. Keep it in mind also, tryptophan. And these are the first 10 amino acids are the essential amino acids. Essential in the sense that they are required by our body in our diet. It means our body can't synthesize these amino acids, so they are needed by our body in the diet. Now let's see the rest amino acids. This starts with YSECQNAGDP. So Y means tyrosine. See, it seems like there is a Y in this name, tyrosine. So Y is tyrosine. S for serine, E for glutamic acid, C for cysteine, which is a sulfur containing amino acid, and also this methionine is also a sulfur containing amino acid. So C for cysteine, Q for glutamine. It seems like cute, glutamine. Q for glutamine. N for asparagine. It ends with N, so asparagine. A for alanine, G for glycine, D for aspartic acid, and P for proline. These are the 20 amino acids, and the amino acid numbered from 11 to 20 are non-essential amino acids. It means these are not required. Not required in diet. It means they can be synthesized by our body, and these non-essential amino acids are synthesized from the essential amino acids which we take through diet. So these are synthesized from these, just structural modification, and we get non-essential amino acid in our body. Now we will see the classification of amino acid. There is a classification of aromatic amino acid. It means there will be a benzene ring in this structure. WYF, W for tryptophan, Y for tyrosine, F for phenylalanine. Just remember this. Then earlier I told you that based on the number of carboxylic group and amino group, the amino acids will be divided into three types: acidic, basic and neutral. In acidic amino acid, the COOH group, the number of COOH group will be higher than the number of NH2 group. And in basic amino acid, the number of NH2 group will be greater than number of COOH group. And in neutral amino acid, the number of NH2 and COOH will be equal. So acidic amino acid, remember, they end with acid, like glutamic acid, aspartic acid, E and D, the symbols. And the basic amino acids are lysine and arginine. We must have heard in that histone protein they are present in high amount, lysine and arginine. So they are basic amino acids. The neutral amino acids are the amino acids which are not included in these two groups, come under neutral group. So you have to remember the DREK, the single DREK, DREK. So except these, all are neutral except D. Now let's see the structure of the proteins. The structure of proteins are of four types: primary, secondary, tertiary and quaternary. The primary structure of the protein shows the most basic and the fundamental structure of protein, where the position of the amino acids can be determined. Like this is the first amino acid, second amino acid, third amino acid, like this. And they are only joined by the peptide bonds, and these proteins are non-functional. The secondary structure, which is a 2D structure, and this structure is formed due to hydrogen bond and also peptide bond. This hydrogen bond is formed between the oxygen of the carbonyl group and the hydrogen of the amide group. And this secondary structure is of two types: alpha helix and beta plated. The alpha helix structure is like a spiral, whereas the beta plated is like a flat sheet. The alpha helix is helical and this one is woven fabric. Alpha helix looks like this and the beta plated looks like zigzag motion. And to remember alpha helix, keep it in mind the structure of DNA, double helix model, like that. The staircases are folded upon each other, stabilized by hydrogen bonds. Then comes the tertiary structure of protein, or the 3D structure. This structure of protein is formed when one polypeptide folds upon itself and it forms many crevices or pockets. And this type of tertiary structure is stabilized by ionic bonds, hydrophobic interactions, hydrogen bond, disulfide bonds, all of these, and also peptide bond. I told you earlier also. Quaternary structure of protein is formed when two polypeptides cross each other and forms pockets. Here you can see an example of hemoglobin, that there are alpha chain and beta chain, both are present in 2. So there are 4 chains in hemoglobin, like 2 alpha and 2 beta. Here is an interesting fact that enzymes are proteinaceous in nature, but they digest protein itself. Like they are proteins and they are digesting proteins. And the proteins can be hormones too, like insulin and glucagon. Thank you. Let's meet in the next video.